All right. Thank you very much, Makoto, for agreeing to give a Build a Cell seminar. And you can get started.
Yes, so then can I start now?
Yep.
Yes.
Yeah.
Then thank you very much, the Kate and the organizers for inviting me to the very exciting activity. I'm Makoto MIYATA from Osaka Metropolitan University in Japan. I'm introducing, the title of my talk is the Motility of the JCVI-syn3, synthetic minimal bacterium constructed by the bacterial actins. This is the outline of my talk. The first, I briefly introduce our interest, in other words, the origin of motility. The second, so then I'll introduce about spiral plasma swimming, which we use the base for the construction of the minimal motility system. So then I'll introduce about minimal motility. So then after that, I'll discuss a little bit about maybe protein, possibility. This is a real-time video of Mycoplasma mobile gliding motility. This is the really unique motility of the unique bacterium. I've been working on this mechanism since 1997. So then we concluded, concluded about the origin of this motility system. So this is a representative video identifying the direct energy source of this motility. Here, they are gliding normally. So then we damage the cell membrane by using the Triton. So then immediately Triton stopped all cells, keeping the cells on the glass surface. So then we removed the DNA and the RNA because the nucleic acids, they work sometimes like the string and inhibit motility. So then we added ATP. In just 5 seconds, they restarted motility, gliding motility with the original speed. So then we concluded that the direct energy source of this system is ATP. So then through all the researches of this motility system, that we concluded this about the evolutionary origin. So then, as you know, ATP synthase is rotating inside of the cell membrane to synthesize ATP and sometimes make the membrane potential. And adhesin is a kind of essential protein for the parasitic bacteria to bind for binding to the host cell surface. So then our conclusion or our suggestion is the occasional contact of ATP synthase and adhesin evolved to be the gliding machinery of this system. Anyway, so then we get to know the story. So then we wanted to overview the motility evolution. This is a kind of it's too much common sense, but the bacteria combination, the bacteria and some kind of the archaea evolved to the eukarya. This is the phylogenetic tree of all the organisms on the Earth based on the information of the genome sequencing. Here are the radiations of the bacteria, and here are the radiations of the archaea, and this is a branch of the eukarya. So that there are so many motility systems, the cell motility systems on the Earth. So then we tried the classification of those motility systems. So that we focused on the structure of the force generating the unit. So then we concluded that all of the motility systems are found published so far can be classified into 18 types. We don't know 18 is the large number or not. Anyway, so then this is the motility evolution along the cell evolution. This is a kind of our idea. But here is the origin of the cell, and they acquired a peptide glycan layer and the DNA segregation. These are based on the cytoskeletal proteins. And separately, bacteria acquired many different types of the motility systems, but all of them are based on the peptide glycan layer. So then these systems didn't transmit to the other, I mean, the archaea or the eukarya. Anyway, we don't know the relation between the bacteria and the archaea, but archaea has only, the one motility system. So some of the archaea, they achieved the membrane remodeling mechanism. So then they became Asgard archaea. So the Asgard archaea finally became a eukarya. So then they obtained the giant cell. And now they can't uh, transfer the material inside of the cell because the cell is too large. So then they acquired cytoskeleton-based transport system. So in some of the archaea, they achieved the motility systems based on the cytoskeletal activities. And then later, they obtained motor proteins like myosin and kinesin and dynein. And the motility inside of the cell that somehow transmitted to the surface of the cell. So then they achieved other types of the eukaryotic cell motility. So then, some, for example, there are animals that they organized the motor protein system. So then they achieved the muscle contraction like this. Then the idea of the origin of the motility, we found something, a kind of, common story. So then that is the housekeeping movements inside. I mean that this system unintentionally, so that they obtained not intended, motility just for the housekeeping, but some of them were amplified by the mutations, and so then the amplified motility was transmitted to the surface of the cell, so then it became the initial motility systems, so then it was selected through the evolution. So then we wanted to discuss this idea based on experiments. So then we remembered interesting system. So that is a synthetic minimal bacterial cell. It was introduced in this seminar system by the Professor John Glass at John Craig Penta Institute. Well, what is the minimal synthetic bacterium, the JCVI-syn3? So it was published in Science magazine in 2016. So that is based on the mycoplasma. I will later explain a little bit more about the mycoplasma. And it's the minimal artificial genome is the feature. And it's featured by the genome manipulation and of course, no motility. So then I'll introduce briefly the spiroplasma swimming. This is again a real-time video of spiroplasma swimming. They look like spirochetes, but actually it's the completely different system. Here are the video images, serial video images, and this is the schematic of the swimming format. But now it's, in this case, the right-handed. So then this is, the tapered end is the front end. So then from the front end to the back side, so then they do the, helicity shifting. So then the shifted part it's the propagate to the back side. And after a while, so then they switch again from the front side. So you think that, the helicity, the switching, so it's the back side of the switching the position. So then it should be the rotated, very rapidly. So then this bacterium pushes cell, the back side. So then they go forward. Here are the electron microscopy of the intact cell. So then we can remove the cell membrane and cytosol. So then we can expose the inside structure. It's called as ribbon. The ribbon is the schematically we can explain the ribbon structure. You can see the gold one, and this is the front side. And actually, this ribbon is the swimming machinery. We isolated this swimming machinery and applied this structure to the electrophoresis, and we found the two classes of the major proteins. The first one is called as fibril. It's a little bit a strange naming, I think. And another the class of the protein is the maybe proteins. So then we isolated the fibril protein and analyzed the structure by the cryo-electron microscopy and clarified this kind of the high resolution structure. And we examined this structure in detail. Actually, this protein was evolved, originated from SAH nucleosides. And we found that the active site is not kept in this protein anymore, like this. So then we also focus on another major protein. So then it's called it's called number B proteins. So it's the classified it's that they have spiroplasma has 5 different types of MREB proteins. And this MREB proteins are involved in the actin superfamily. So then here are the structures, the 3D structures of the MREB proteins. And you can see the actin protein. So then you can see the, clear similarity, of these structures. Anyway. But MREB protein belongs to the actin superfamily, but they are not involved at the motility at all. Actually, the MREB protein is a kind of the ancestral protein of actin, and they form a kind of the short filament. It's the responsible for the, it has a role of the scaffold for the peptide glycan synthesis complex. So then I'll talk about the minimal the motility. It's the kind of synthetic biology. Here is the spiroplasma genome. So then on the genome, there are these proteins, candidate proteins of the uh, swimming machinery components are coded something like this. So then we intended to integrate these genes into the single DNA fragment. So then express these proteins in the SYN3. And then we see what happens. We assembled these proteins and, all the directions. And so then we express these single DNA proteins, including the 7 proteins we expressed. So then see what happened. But here are the representative video of this minimal swimming motility system. So then the leftmost panel is that this is real-time video again. It's a kind of the goal. It's the real spiroplasma swimming. The middle is the SYN3. It's just the most cells are just spherical. And so then the cell size is not so much very uniform. Then they can move just only by the Brownian motion. The rightmost one is the swimming SYN3, harboring the 7 proteins. They are, doing the helicity shift, and sometimes they swim to the one direction. And we analyze the dimensions of the helical structure. So then this is the width of the helix. So then regarding the original spiroplasma, the number of the 0.5 something and the swimming SYN3 was similar. And the pitch of the helicity, so it was regarding the spiroplasma, it was 0.68, and the swimming SYN3, it was 0.63. It's not so much different. And also we analyzed the structures of the spiroplasma and the swimming SYN3 by using the cryo-electron tomography. So then here the cell membrane, and you can see it's a little bit difficult, but not obvious for the non-electron microscopy people, but we can see the clearly. So then we see that filamental structure, and also that we analyze the motility in detail of the spiroplasma and the swimming SYN3. And these are the half the speed of the videos. And we cut the frames and the straighten the cell images like that. So then we trace the helicity switching. So then you can see, here, so then the spiroplasma, regarding spiroplasma, so the moving of the spiral helicity shift was the speed of the 8.8 micrometer per second. So then if we calculate, convert to the rotation speed, it was 12 per second. So then regarding the swimming SYN3, the speed, shift speed along the cell axis was the 8.2, and the rotation speed was this number. So that these are not so much different from original spiroplasma. So then it suggests that we somehow reconstituted the swimming, healthy shift swimming motility in SYN3. But you can notice that somehow, different. That's we trace the, right-handed position and left-handed position. We colored like this. So then the spiroplasma is the, helicity shift moving is very smooth, continuous. But the swimming SYN3, it's not continuous. So that it easily that they they stop and they, reverse the reverse the, the moving direction. Anyway, so then the discontinuity is reflected by the swimming behaviors. So then if we trace uh, the, the front side in the swimming behaviors for in this graphs, we we showed the 10 cells. So then you can see the spiral plasma cell that go to the distant place in 10 seconds, but swimming since that frequently they change the swimming direction, so then it's impossible to go to the distant place. So it may be related to the structure, because if we see the spiral plasma, you can see the tapered end, but we can't see any tapered end regarding the swimming symmetry. So that may be the one reason for the discontinuous, swimming behaviors of the swimming symmetry. So then next, we focus on the, roles of the individual the proteins. So then we removed, we stopped the expression of individual, proteins genetically. So then this is the spiral plasma original cell the video, and this is the swimming syn3, and this is the original just syn3. So then we removed the individual the proteins, but unfortunately, so then the cell appearance didn't differ from the, normally, what is normally, and from the 7 genes, swimming syn3. And just only the maybe 5, the the dimensions of the helical dimension is obviously different from the 7 genes one, but still they were moving somehow. So then we concluded that probably maybe proteins have the redundant roles. So then we changed our the thinking. So then we tried, the bottom-up expression. So then we expressed the individual, the single, the proteins, but all of them didn't give the motility. So then we tried, the pair of the Mrevi proteins. So then we analyzed DNA sequences of Mrevi proteins. So then we classified 5 Mrevi proteins into the 3 groups. The first group is I don't discuss the too much detail, but they were classified 5 and 2 group, and 4 and 1 group, and the 3, the class of the 3. So then we thought that, if they are redundant, so then if the important proteins maybe have the, two, the class of including the 2 proteins should have the, critical roles for the swimming. So then we try first, combination and 5 and 4 and 5 and 1 and 2 and 4 and 2 and 1. So the result was something like that. So then combination and 5 and 4 and 5 and 1. So then the behavior and appearance of the swimming cells were similar to the 7 genes ones. And the 2 and 4 and 2 and 1, they gave, some helicity and some movements. So the other combinations, some of them showed helical structures, but no movements. So then these are all the Brownian motion. So then our conclusion is something like that, regarding the origin of the motility or something like that. Mrevi is, as you say, Mrevi is the widespread protein in the bacteria. The Mrevi forms, kind of the short filament then regarding the evolution, so then okay, somehow that they accumulated the mutation. So then the proteins it was classified, evolved to the different classes. So then somehow that they got the more stable the structure. So then the interaction between the two proteins may have some, the fourth generation. So then they can do, the conformation change, formation change of the filament. So then depending, ATP energy depending, the switching may cause the conformation change, switching of the helicity. So then they give some motility. So then regarding the experiments, we achieved the minimal motility system because we don't know anything, you know, uh, which can give the, cell motility with this small size of the proteins. So then we can say that, we achieved the minimal motility system on the minimal, the bacteria. So then regarding the evolution, so then they somehow selected and somehow, the system was supported by the more differentiated maybe proteins and also the other proteins supporting the ribbon, for example, fibril proteins. So this is, the story of the evolution, start of the swimming motility of the spiroplasma. Now, this was the paper, describing this story obtained, 300 of the autometrics. So then for me, this is the first, experience in my life. And also the big, my, surprise was that our paper was focused by the Scientific American as the one page of the column. So then because of I've been, uh, enthusiastically. So then I love the Scientific American. So then I continued to read it 40 years. So then I was very happy about that. Anyway, so then I will discuss more about the performance and the possibility of the MureB protein. So as I told you, the MureB is one of the actin, the superfamily proteins. So then, and also very, very interestingly, the only, the mycoplasma, in other words, the molicutes, the class molicutes, sometimes we call it as the mycoplasmas. So then only, this phenomenon can be found in the mycoplasma. So then what is the features, of the mycoplasma? So it's the genome reduction and the loss of the peptide glycan and the use of the cholesterol and the use of the cytoskeleton. So then recently, as you know, so there's so many, genome information, including the metagenome, are available. So then we are remodeling the phylogenetic tree of the mycoplasma. So then mycoplasma is featured by the small genome. So then, but it is, the genome reduced by the parasitic lifestyle. So then actually it is the, original, normal, what is normal? Original bacteria was bacillus. So then if we make the boundary, here are the world normal, kind of normal bacteria, and here are the mycoplasma group. And recently, the boundary and here are the spiroplasma and the syn3. And the syn3 is originated from the mycoplasma mycoides. Anyway, this is the kind of well-known mycoplasma are here. And also the very, very interestingly, recently, so the the very ancestral Mycoplasma species, Mycoplasma the genus, have been identified. And very, very interestingly, so they are not the parasitic anymore. So then it's a kind of mystery anyway. So then we looked for the other maybe proteins in the Mycoplasma groups and found in the boundary, very ancestral Mycoplasma species, we found new maybe groups. Here are the, bacillus, the maybe proteins here, and here are the spiroplasma maybe proteins. And here are, this is we believe that currently that this haloplasma thing is, the most closely related to the bacillus. So then, interestingly, bacillus, the haloplasma have as many as the 7 maybe proteins. And also the plasma is also the related to that. So then we focus the haloplasma. So actually, haloplasma was isolated in the 2008, and it's the anaerobic bacteria deep sea. But we can see some, the moving video like that. And here are the same images. And we determined the genome and concluded that 7 maybe the proteins are coded on the genome of the haloplasma. And we tried, the different many, combinations of the protein expressing in the, the SYN3. We tried the different combinations of the haloplasma molecules, in the SYN3 as we did in the spiroplasma. So interestingly, if we express that, so these all the not actually we eliminated the great things, but if we express the 7 maybe the proteins, so that they gave very strange interesting motility. This is also the real-time video. And just the 1 and 2 that they gave the this coiling motility, and 3 and 4 that they gave. This is a kind of one pattern of the coiling motility, but it's the discontinuous, the curving, the motility they showed. So then, as you can see, the haloplasma, the motility is it's the curving or coiling. So then, basically, they are in the straight, the filamental structure, and so they keep the coiling, and sometimes the coil moves along the cell axis. Now this is completely different from the spiroplasma, because spiroplasma can move only when they only after that they form helicity, and so then if they form the helical structure, so then they never convert to the straight and helical, but instead they do the handedness the shifting, right-handed and left-handed. So then it means that somehow in the mycoplasma groups, the maybe is now somehow easily convert from the non-motile to the the moving, the swimming protein. So then now we believe that we are just seeing just after converting the static short filament maybe to the somehow moving protein. So then it is that we are now seeing this the boundary from housekeeping movements inside to the amplified initial motility. So that is our story. So then this I should present acknowledgement. This the story was done with the really nice the collaborators outside of our lab. So then I list our friends here, and the most parts were done in our lab in Osaka Metropolitan University. And so then Dr. Kiyamahana, so then this person, she, developed the SYN3, the cell biology, the system, and found the really exciting spiral plasma stories. That's it. Thank you for your attention. Any questions are very welcome.
Thank you. Thank
Thank
you
you.
so much. Oh, I think my camera was off. Thank you so much. There are several questions in chat. Can you see chat?
No, how can I see that? Sorry, no, I can't see that.
I can read the questions for you if that
Yeah.
helps. All right. So the first question is from Henrik. This might be a naive question, but if you knock out genes responsible for motility and previously mobile cells become immobile, how will the cells feed? Don't they need to move to seek nutrients?
Sorry. So is that question about the original the species or the on the SYN3?
Based on the time in which it was asked, I don't know, Henrik, you can unmute yourself if you want, but just based on the timestamp I see, I think it's about the original experiments.
Yeah, you mean that the if we knock out the motility the proteins in the spiroplasma, but that is that the question you think?
That's my guess.
Yeah,
I
yeah.
can.
Then I probably I can read it. Right. Yes. So then actually for us we we should be able to knock out the spiroplasma, but actually not so many people are working on the gene manipulation of the spiroplasma. So then actually, so then we can't currently, that we can't knock out the spiroplasma of the genes, freely. But some people that publish the mutant, deficient for the MREB5. So that of course, if without MREB5, they can't form I don't remember it that they can't form the helical structure. So then that's true. So then if we knock out the MREB protein that they can't swim. And actually, we were trying to develop the spiroplasma, the gene manipulation system, but actually we tried also the synthetic cell, and the synthetic cell it worked much more than the spiroplasma. So then we shifted to the synthetic SYN3 experiments.
Thank you. Okay,
Thank
so
you.
next question. Great talk. Thank you very much. Would it be possible to reconstitute actin cytoskeleton in vitro in vesicles and see the cells start swimming?
Yeah, so it's really nice question. So, but we are, and also our friends are trying. But the one, the problem is that we try to purify isolate the proteins, and the MolB5 is kind of easy. But regarding other proteins, it's difficult to get soluble the fraction in the E. coli. So then we are trying the different ways. But some people are trying, but the getting the soluble protein from E. coli is kind of difficult. So then our guess is that some, the folding the Murebis may need the chaperone, and the chaperone, the specificity of the E. coli may be the difference from the spiroplasma. So then actually, as I told you, the SYN3 it is the kind of the relative of spiroplasma. So that's the reason why we successfully express the spiroplasma the proteins in SYN3. And another, the possibilities we made, use the proteins isolated from the SYN3. But the SYN3 has some the problem about, the growth, the yield of the cells. It's the completely different from the E. coli. So then we are, improving, from the both directions.
Thank you. That was awesome. Actually, I see there's a Angelica has a follow-up question. Says, do you think it would be possible to engineer a different motility system, something
Right.
engineered from scratch, like DNA origami based, or with other proteins, something
Right,
orthogonal to
right.
actin?
That's what we are doing.
Yeah.
Recently we are trying the mutations and the combinations, the artificial combinations, maybe expressions. So then somehow that we are getting some, the good feeling about the unusual, the structural change, and also the combination of the heterogeneous, the maybe proteins, and also the combination of the fibril and the other combinations and the mutations. So then we are trying completely trying to make and the completely new cell shape and the new the motility. Yes, that's a really, exciting field.
Yes, I'm really excited to see that too. And I see last question in chat from
Go
Chris.
ahead.
Great talk, thank you. Do you think we could use the new cell-free system from Mycoplasma to express the cytoskeleton proteins? And will they assemble into cytoskeleton?
Yeah. That's also yes, yeah. So I think I haven't read the successful the papers, but and some people are working on that. So then we want to try that. So then we are trying the different, the different the directions. So then to use the E. coli, of course, and to improve the growth to the isolate the protein from the SYN3. And another possibility is that we may use the original mycoplasma. So then the mycoplasma mycoides is kind of we can get the good yield or something like that. So then I agree.
Yep. All right, thank you. Thank you so much. Yeah, I just want to say for myself that I'm really excited, especially for the orthogonal engineered cytoskeleton work. I think that's going to teach us a lot about the evolution too, once you kind of map out the boundaries for it. Okay, if anyone has any other questions, last chance. If not, I want to say thank you very much, Makoto, again for a
Yes,
fantastic
thank you very
talk.
much. Yes, it's my great pleasure, our great pleasure.
And thanks everyone, and have a great rest of your week. Bye.
Yes, thank you. Good night. Good morning. Good night, people.
Bye.